van Iersel has been with the University of Georgia's Department of Horticulture since 1995, where he now holds a Dooley professorship. His research focuses on cost-effective supplemental lighting technologies in greenhouses and vertical farms. He's the director of Project LAMP. It's a $5 million US-based research project that brings together plant scientists, engineers, and economists to develop profitable supplemental lighting strategies. In 2017, he co-founded Candidas Incorporated to help bring novel lighting strategies to the greenhouse industry, and he's published over 130 scientific papers and has given invited lectures about his research around the world, including Italy, Spain, Taiwan, Canada, Chile, Brazil, and now Massachusetts. So I'll let you take it from here.
Okay, thank you very much, Carrie. Let's see if I can get the screen set up properly. Hopefully you see my PowerPoint right now. I don't think you see my video, which is okay. But you do see the PowerPoint?
Not yet.
Not yet.
Let me try to share screen again.
Right now we're just seeing your face.
Yeah, so you're much better off seeing the screen, I can promise you.
Yeah, there it is, Mark.
Okay.
Perfect.
Thank you so much, and thank you for the invitation to talk about the research that we do in my lab. Let me see if I can minimize that bar at the top of the screen. I'm not even, I assume you see that too, so I should hide that. Okay, that's better. And so I have really two objectives today with this presentation. One of those objectives is to talk about the research that we have done with red, green, and blue light, and hopefully clear up some misconceptions that seem to be widespread with regard to their efficacy related to photosynthesis. And I want to start out by giving all of the credit for this to June Liu. June is a PhD student in my lab, and she said that she would be happy to answer any of the difficult questions that you may have. She is on the call today, but the data that I'm showing here today are hers. And so the first thing that I want to do today is to basically just talk about those data. But the second objective today is going to be to actually talk about how you can use your leaf gas exchange data and get the most possible information out of that data. If you are going to spend a lot of time doing these kinds of measurements, you might as well also then spend the time on trying to figure out exactly what it all means. And so with regard to leaf photosynthesis and spectra, there are a lot of misconceptions out there, and you can find some incredibly bad information on the internet. And a lot of that information is based on this chlorophyll absorbance spectrum that you see right here, which shows that chlorophyll is not very good at absorbing green and yellow light. And as a result, you can find information on many websites that says that plants cannot use green and yellow light for photosynthesis. That information seems especially prevalent on websites from lighting manufacturers who make LED lights that have red and blue LEDs built into them. If you go to the website of lighting companies that make white LEDs, they typically prefer this diagram, which is also becoming pretty common online. And so this diagram is pretty interesting because it actually shows that there are two pigments that absorb green and yellow light pretty well, phycoerythrin and phycocyanin. And if you've never heard of those, that's fine, because I would strongly recommend that you forget about them again as quickly as possible if you are working with higher plants, because those pigments are not present in higher plants, are present in cyanobacteria. So when it comes to higher plants and photosynthesis, there are no pigments that actually do absorb yellow and green light very well. So with regard to photosynthesis, it largely comes down to chlorophyll and carotenoids that are present in the chloroplasts. And as you can see here, none of those pigments are particularly good at absorbing anywhere from 525 to 625 nanometer light. But we need to keep in mind that these absorption spectra are always collected in extracts with organic solvents, not in intact plants. And it turns out that absorptance really changes depending on what you're measuring. So this diagram, which is based on a really old paper from 1952, shows the absorptance spectra for, at the bottom here, the pigment extract, the solid green line, and it shows that in a pigment extract, yes, green light is actually absorbed pretty poorly. But if you extract chloroplasts, instead of extracting the pigments from the chloroplasts, it turns out that absorptance is much higher in this part of the spectrum. And if you look at whole leaves, you will see that even at the wavelengths that are absorbed the least, you still have about 70% absorptance. But it still raises the question of why did evolution come up with a pigment composition on leaves that seems to not be very good at using the entire spectrum? Is there a reason for that? Generally speaking, evolution isn't very forgiving of inefficiencies. So keep that question in mind as we talk about different colors of light and their effects on photosynthesis. So the effects of different colors of light on photosynthesis have been studied for a long time. This is perhaps the most well-known study done by Keith McCree, published in 1971, where he shows the relative photosynthetic efficacy or quantum yield of different colors of light. And this is a study that, best I can tell, is cited a lot and read very little. It is often miscited. People don't seem to really understand the limitations related to the work that Keith McCree did, even though Keith McCree himself pointed those limitations out very clearly. So one thing is that he did not look at interactive effects of different colors of light on photosynthesis, and there are very strong interactive effects between 400 to 700 nanometer light and far red light from 750 to 750 nanometers. But more relevant for what I want to talk about today is the fact that he collected all of these data under very low photosynthetic photon flux densities. And that is fine, but that limits the conclusions that you can really draw from this work. And people often citing this work will say that green light has a low photosynthetic efficacy. But when you look at the actual curve and you compare green light to blue light, it actually turns out that those are very similar. Well, people will always say that red light is used most efficiently, while in reality, it's really orange light that is used most efficiently. But related to the limitation that this research has with being done at very low light levels, in 2009, Terashima et al. Published a very elegant paper where they looked at what they called the differential quantum yield of CO2 fixation. And that is the increase in photosynthesis that you get from applying additional light. And in their case, what they did is they developed a light response curve with white light, which is indicated in this graph right here. So these are the photosynthetic rates of white light in response to increasing light levels. And then what they did is they added either red or green light to that white light, and they quantified how much red and green light increase photosynthesis. And when they did that, to start off with, in the absence of any white light, what they showed very clearly is that red light here increases photosynthesis more than green light does. And that is entirely consistent with Keith McCree's work. So this wasn't particularly novel, but what they also showed is that if you add red and green light to a background of high white light, it turns out that green light actually increases photosynthesis more than red light does. And that is the opposite of what you would expect based on the work by Keith McCree. What they showed was that higher differential quantum yield of green light at high background white light is caused by the ability of green light to penetrate more deeply into leaves. And green light can do that because it is absorbed less efficiently. So actually the low absorptance of green light provides the green light an advantage here. And that is illustrated in this image that you see right here. These are false color images that show how deeply different colors of light can penetrate into a leaf. And so the dashed lines that you see show the top and the bottom of the leaf. And it is very clear that the red and blue light don't penetrate very deeply into the leaf. And that means that only the palisade parenchyma cells can actually photosynthesize, because the spongy parenchyma underneath just doesn't receive any of that red or blue light. But when you look at the green light, you see that light penetrates much more deeply into the leaf. And as a result, those deeper cell layers can also contribute to overall leaf photosynthesis. And so in our study, we actually quantified how efficiently different spectra are absorbed. And so with the light we used, the leaves absorb about 92% of red light, 81% of green light, and 93% of blue light. But that low absorptance of green isn't necessarily a big drawback if that green light can actually efficiently stimulate photosynthesis in more cells than red and blue light can. So when we started this research out, just to be contrary, what we started off with as a hypothesis is that green light can drive photosynthesis more efficiently than red and blue light, but especially at high ppfd, when red and blue light saturate those upper cell layers but do not excite the lower cell layers in the leaf. And we were also interested in looking at how efficiently these different colors of light actually stimulate photosynthesis based on the amount of absorbed rather than incident light. So in the study, we used green Taurus lettuce plants, which you can see right here. They were grown inside of a growth chamber, 14-hour photo period, fluorescent light at 100 micromoles per square meter per second. And then we did our photosynthetic measurements using 3 different colors of LEDs. These are LEDs that are built into the LED unit that comes with the Cyrus 3 leaf photosynthesis system. So we had blue, green, and red light. And when you look at the spectrum on the right-hand side, what you see is that based on the McCree curve, you would expect the green light and the blue light coming from these LEDs to result in the same photosynthetic rates, while red light would be expected to have substantially higher photosynthetic rates. So all of the data were collected using a Cyrus 3 leaf photosynthesis system using the light unit that you can get to put onto the cuvette. And June did light response curves on 9 different lettuce plants. She used 9 different spectra total that included pure red, green, and blue, but also all kinds of combinations of red, green, and blue. I'm not going to talk about the combinations of red, green, and blue because it really didn't show anything particularly interesting. And the data were collected at ambient CO2 concentrations because the plants were grown under ambient CO2 concentrations. So to show you the light response curves, give you a quick idea of what June actually found. You can see pretty clearly in this graph that at low light levels, as would be expected based on the McCree curve, red light results in higher photosynthetic rates than green or blue light. But when you look at high light levels, and this is more consistent with the findings from Terashima, we actually see that green light results in slightly higher leaf photosynthetic rates than red light, and overall blue light just performs pretty poorly compared to red or green light. once we had these light response curves, what June did was fit a curve through those data, and the curve that she used was an exponential rise to a maximum. We typically use this equation partly because of its simplicity. And I hope I'm not muted right now. I am. Thank you. I'm unmuted again. What I like about this equation that it has 3 regression parameters, and they have a very clear physiological meaning. So the dark respiration is simply the y-intercept. The PG max is the maximum gross photosynthetic rate that the leaf can achieve under light saturating conditions. And the B parameter is used to calculate that initial slope of this curve, which is really the same thing as what Keith McCree did, the maximum quantum yield that can be achieved. And we typically use this regression equation because it's simple. It has 3 parameters with a clear meaning. And based on what we see, it typically fits the data very well. One thing that you will often see in the literature is this equation right here, which has a 4th regression parameter, that is the theta. And that theta describes how quickly this light response curve curves when you increase PPFD levels. But it has, best I know, not been tied to a very clear physiological parameter. And so I sometimes refer to it as a fudge factor. So if you can get a good fit with fewer parameters, then that's my preference. So once we have these light response curves, once we have the regressions, we can very easily calculate that maximum quantum yield of CO2 assimilation. Again, the same thing as what Keith McCree did in his study. And so that's just the initial slope of that curve. And so when we look at the maximum quantum yield for different colors of light, what we see is that blue and green light have a lower maximum quantum yield of CO2 fixation than red light. And this is based on the amount of incident light. And these findings are completely consistent with the findings from Keith McCree and others who did similar work. But we were also interested in seeing what the quantum yield of CO2 fixation is if you look at it based on the number of absorbed photons. And because green light is absorbed less well by these plants than blue and red light, that changes the results a little bit. And so when we look at the same measure, maximum quantum yield of CO2 assimilation based on absorbed rather than incident photons, we actually see that green and red light have a similar maximum quantum yield, while blue light has a lower maximum quantum yield. And so what this tells us basically is that there is nothing wrong with green photons with regard to photosynthesis, other than the fact that they are not absorbed as well as red photons. But if green photons are absorbed, they're perfectly capable of efficiently driving photosynthesis. Blue photons, on the other hand, look relatively poor. And the reason that blue photons have a lower maximum quantum yield is that blue light is partly absorbed by non-photosynthetic pigments, especially anthocyanins. And if anthocyanins absorb some of those blue photons, then that light cannot be used for photosynthesis. In addition, carotenoids absorb some of the blue light, and they are not as efficiently at transferring that excitation energy as chlorophyll is. So we lose some efficiency there as well. So going back to this curve, the other thing that we determined from these regression curves is the maximum gross photosynthetic rate, which is the vertical dashed line on the right side of this graph. And when we look at that, things get really interesting in my mind, because what we see here is that the maximum gross photosynthetic rate is actually highest with green light, followed by red, and blue is still at the bottom. And so this dispels the notion that green light cannot be used efficiently for photosynthesis. It certainly can be, but especially at high light levels. And the reason for that, once again, being that this green light can simply penetrate more deeply into the leaves and therefore excite chlorophyll in more of the chloroplasts as compared to red or blue light. So there are some other things that we calculated from these light response curves, and one of them is the actual quantum yield of CO2 fixation. So I've talked about the maximum quantum yield, which is the initial slope of that regression line, but the quantum yield of CO2 fixation depends on the amount of light you actually provide to the leaves. And to illustrate that, so at low light levels, you can see the slope of the line that just appeared in the graph is the quantum yield in moles of CO2 fixed per mole of photons. But if you calculate that same parameter for higher light levels, you can see that the slope of that line decreases, meaning that the actual quantum yield of CO2 fixation decreases with increasing light levels. And so June actually calculated this for different light intensities based on the actual data she collected, not based on the regression curve that she developed. And then you get a curve like this that shows very clearly that, yes, at low light levels, red light does have a higher quantum yield of CO2 fixation than either blue or green light. But at high light levels, green actually has the same or perhaps even a slightly higher quantum yield of carbon assimilation as compared to certainly blue light and perhaps even red light. So this once again shows that green light, when applied at high ppfds, is actually used relatively efficient compared to other colors of light. And then the last parameter that we calculated from these light response curves is equivalent to what Terashima et al. Did, the differential quantum yield. And what that is basically just the slope of this regression curve at different ppfd levels. And when we look at that, we first of all see very clearly that the differential quantum yield decreases very rapidly with increasing ppfd. So basically what that means is that if you have a high amount of light already, providing even more light doesn't have a whole lot of effect on leaf photosynthesis. If we look at these data in more detail at low ppfd, what we see is that the differential quantum yield, or the increase in photosynthesis with an increase in light, is highest for red light, followed by green, and lowest for blue light. But when you get to ppfds of about 230 micromoles, the green light actually starts having a slightly higher differential quantum yield than even red light does. And when we blow up the part of the graph at high ppfds, as you see right here, first of all, I want to point out that all of the values now are much, much lower than they are at low ppfds, but you can also very clearly see that the differential quantum yield of green light is much higher than that of red or blue light. So at high light intensities, supplying additional green light has a bigger effect on photosynthesis than supplying additional green or red light. And that is completely consistent with the findings by Terashima. So we wanted to better understand why we see these differences, and to do that, we also measured CO2 response curves. And from CO2 response curves, you can quantify 3 processes that may limit the overall rate of photosynthesis. And that is shown in this diagram here from a paper by Sharkey et al. It's kind of an unusual way to show the photosynthetic pathway, but it's really useful in this context. So on the left side of this diagram, you see that Rubisco can limit photosynthetic rates, and that is the case normally under low atmospheric CO2 levels. As CO2 concentrations increase, at some point, the ability of the leaf to regenerate the amount of RUBP that is used up by Rubisco can become limiting. And with CO2 response curves, that is often expressed as the electron transport rate that is required to regenerate the ribulose bisphosphate that is used up by Rubisco. And then at really high CO2 levels and high light levels, what may become limiting is actually the ability of plants to use the triose phosphates that are being produced in the Kelvin cycle. So June did CO2 response curves, and more specifically rapid ACI response curves at two light levels, 200 and 1000 micromoles. That's a bit unusual to do these curves at low light levels, but we had a really specific reason to do that, because when we look at the light response curves and we look at 200 micromoles of ppfd, at that light level, we see that red light is used most efficiently, followed by blue, with green being used least efficiently, while at a ppfd of 1000, the order of these different lights, of these colors changes, with green having the highest photosynthetic rate, red just below it, and blue the lowest. And we wanted to understand why that change occurs in response to higher ppfd levels. So from the rapid ACI response curves, June calculated the electron transport rate that is needed to regenerate the RUBP that is being used up by Rubisco. And she also calculated the maximum Rubisco carboxylation rate. If you want to learn more about these rapid ACI curves, you should go to the PP Systems webinars page, because just a few weeks ago, Jim Bunce gave a webinar on this specific technique, so I'm not going to go into the details of this. Instead, what I want to show you is the results that June got. So at low ppfd levels, what we saw was that the electron transport rate is highest under red light, followed by blue, and lowest with green light. And that is completely consistent with what we saw in the light response curves, where red light at 200 micromoles has the highest photosynthetic rate, while green light results in the lowest photosynthetic rate. So it was really nice to see that these results back up the findings from the light response curve. And then at high light levels, 1000 micromoles, first of all, we see that the electron transport rates are much higher, as one would expect with more light. But now we see that green and red light have very similar electron transport rates, but blue has much lower electron transport rates. And once again, that is completely consistent with our findings from the light response curves. And so June also calculated the maximum carboxylation rate under high and low ppfd for these different colors of light. And what we saw there is that the maximum carboxylation rate is much higher with high light than with low light, but we saw no effects of the light spectrum on the maximum carboxylation rate. And so from all of this work, June and I were able to draw some conclusions. And first of all, so we did see very clear interactive effects of light color and light intensity. And specifically what we found was that under low light, green and blue light do result in lower photosynthetic quantum yield, lower photosynthetic rates than red light. And as I mentioned before, the reason for that is that blue is at least partly absorbed by non-photosynthetic pigments, while green light is not absorbed as well as red light, resulting in lower photosynthetic rates. But when we look at the photosynthetic capacity of leaves under high light, now the picture changes. And this, I think, is where things get really interesting, because we were able to show that under green light, leaves can achieve higher photosynthetic rates than with red or blue light. And so that is really different from what one would expect based on the McCree curve. And I want to make it clear that I am not criticizing Keith McCree here in any way, because he makes it really clear that his findings are limited to very low ppfds. We just need to make sure that when we cite that paper that we include that limitation in our discussion of the results. And so why did these results occur? Well, what we were able to tell from the CO2 response curves is that the effects of light quality on photosynthetic rates result from effects on electron transport rate. For some reason, someone, I don't know who, thought that J would be a good abbreviation for electron transport rate, probably because the letter J does not occur in electron transport rate. But so it makes perfect sense that different colors of light, that they would affect the light reactions of photosynthesis. We did not see any effect on Kelvin cycle, specifically Rubisco activity from different colors of light. And as I mentioned briefly earlier, June also did all kinds of combinations of red, green, and blue light, and we didn't see any kind of interesting synergistic or antagonistic effects when we combined red, green, and blue light. With that, I would first of all really like to thank June for doing all of this work and allowing me to present it. This work was done as part of a USDA-funded project called Lighting Approaches to Maximize Profits. As part of that project, one thing that we are trying to do is gauge the impact of the work that we do, and related to that, you will soon respond a follow-up survey from a graduate student working on this project, Amanda, and hopefully you will take the time out of your schedule to actually respond to that survey. It shouldn't take a whole lot of time. And with that, I hope we have time for questions, and I hope that June is ready to answer all the difficult questions. Thank you. And I'll see if I can
thank you, Mark. Tim, are you going to, Tim has some questions that have come through the chat, and he was going to be bringing those to your attention.
Yeah, so Mark, Karina would like to know how long the plants were grown under the light treatments before you did the light response curves.
So these plants were never grown under the light treatments under which we collected the data. The reason for that being pretty simple. You can't grow these plants under just red, green, or blue light. They do need pretty much a full spectrum to grow normally. So they were grown under white light, and then just for the measurements exposed to the red, green, and blue light.
Right. Selena would like to know, do light quality effects on leaf anatomy affect the photosynthetic responses that you presented today, where plants grown under white light will have a different anatomy than those grown under dichromatic red and blue light or monochromatic red or blue light, and will changes in the leaf anatomical structure drive different gas exchange responses?
I have no doubt that they do, but that relates to the previous question. We didn't grow these plants under different spectra. We have grown the same cultivar of lettuce under just red and blue light, and you wouldn't even recognize the cultivar. The entire morphology of these plants changes, and I'm quite certain that that affects the physiology as well, but we haven't looked at that specifically. And growing the plants under just red, green, or blue light, once again, I don't think that the plants would actually be able to survive.
And a follow-up question to that is, what type of responses can we expect when using white or dichromatic light as a light source? There are a couple of studies that have shown that when using red and blue LEDs in light sources, photosynthesis can be higher than that measured under ambient light that's used during plant growth and development, or under monochromatic light, similar to what you showed in your slides.
Yeah, that's a really complicated question. I'm not sure that I have a very satisfactory response to that. So once again, I mean, growing these plants under single color light is really difficult. When you grow them, at least this particular cultivar of lettuce, when you grow it under red and blue light, which is common in LED light sources, they grow very strangely, which undoubtedly, again, impacts their physiology. We don't have the setup here to grow plants for long periods under a wide range of different spectra, but quite a few plants do really need red, green, and blue light, or a full spectrum, to be able to have what I would consider to be a normal morphology and presumably a normal physiology as well, but that's about all I can say.
Right. How much do you think these results can be generalized to other plant types?
I think that with pretty much all species, you would find very similar results. We haven't done that, just doing all these light response curves and CO2 response curves on one species took a lot of time, but the photosynthetic mechanism of different plants is very similar. And so I would expect that you would see very different results. Very similar results.
Right. Was there any data collected related to the growth and yield, and how is that connected to your physiological data?
No, so once again, I mean, the plants were all grown under the exact same conditions. So there was no point in doing growth or yield. But I'm very appreciative of that question, right? I talked a lot about photosynthesis, and I could give a whole other talk about why leaf photosynthesis is in many cases a really poor indicator of plant growth. So you cannot correlate in many cases high photosynthetic rates with rapid growth. There are many other factors that affect plant growth, with the most important factor probably being the size of the canopy. And so you can manipulate the light spectrum to get plants to develop a larger canopy, allowing them to intercept more light, and they will grow faster as a result. And that effect on the plant morphology with regard to growth is probably more important in the vast majority of cases than the effect of light on the actual photosynthetic rate.
Right. So another question is, how can we leverage the light treatments for quality parameters of fruits like color, shape, and taste?
There has been a lot of work on leaf color, I think less so on fruit coloration, and I'm not aware of much of any work on shape. There has been some work on taste as well. And so you can use the light spectrum to manipulate secondary metabolism. And generally speaking, the more blue light that you include in the light spectrum, the more secondary metabolites accumulate in the plant, whether that is anthocyanins, in many cases flavor compounds, or cannabinoids. They all tend to increase with an increase in blue or UVA light. So there is lots of really fascinating opportunities there.
Right. So you mentioned the plant needs a full spectrum of light for growth. Do you have a recommendation of the combination of using red, green, and blue light?
So I think most plants, but this is species and even cultivar specific. If you have, some plants will grow just fine under just red and blue light. The plants that do not, typically if you mix in just a relatively small amount of green light, I think they tend to do well. But I think we actually have, I know we have some people from NASA on the call. They may have done more work on that than I have. But I guess they're muted, so they can't chime in.
Yeah, I mean, we can probably get responses to that if we need to. Another question is, what would be the mechanisms behind the response of electron transport rate to light quality? Any idea about the effects over reaction centers?
Yeah, so what we assume is the case that, so with red and blue light being absorbed so efficiently by the top probably 2 cell layers in leaves, you have all of that excitation energy in those chloroplasts that is getting funneled towards the photosystems. And as a result of that, those photosystems are going to be closed relatively much, and the plants have to upregulate non-photochemical quenching or heat dissipation in those cells, and that is going to reduce the overall amount of light that can actually be used for the light reactions. But green light, especially because it isn't absorbed as well, and therefore can penetrate much more deeply into the leaves, is much less likely to oversaturate the light harvesting complexes and reaction centers in the upper part of the leaf, because those cells simply do not get as much light as they do with red and blue light. So that allows the reaction centers to be more efficient, and you don't have to have the same amount of upregulation of heat dissipation as you do with red and blue light. So that gets me back to the point that I made earlier with regard to evolution. And I think evolution came up with a very efficient solution here by allowing part of the light spectrum to penetrate more deeply into leaves. I have no doubt that evolution could have figured out a pigment that is really good at absorbing green light and using it for photosynthesis. But if you think about a leaf that would have a pigment that absorbs all of the photosynthetic light very efficiently, that would essentially mean that only the top 1 or 2 cell layers in a leaf could photosynthesize. And by allowing some of that green light to penetrate more deeply, now you actually have the entire leaf contributing to photosynthesis. So I think that we're looking here at a nice evolutionary mechanism that increases the overall photosynthetic efficiency.
Right. Another question would be, did you test potential differences in response of C4 species?
No, we have not done any work with C4 species, partly because of the USDA project that is funding this work is focused on horticultural crops, and we don't have a lot of economically important C4 species, especially crops that would be grown in controlled environments. So we have not. Would be interesting to do though.
Right. Another question would be, how does a lettuce grower use this information with regards to green wavelengths? Lettuce has been shown to use light more efficiently at lower PPFD. Do you see growers exposing plants to high PPFD without worry of adverse physiological effects?
So there are a few components to that question. So first of all, with regard to the light spectrum, I don't think we need to be as afraid as we used to be of white light compared to red and blue LED light, because the white light with a substantial amount in the green part of the spectrum is going to be used just fine by that crop. You're not wasting that light. So I think the idea that you have to have red and blue light is outdated. There are some efficiency advantages to red and blue LEDs over white LEDs, but from a physiological perspective, not so much. But the other thing, so what you see in this data set is that plants use light much more efficiently, as you can tell from the quantum yield, at low PPFD than at high PPFD. So one other important take-home message of this is that if you are going to provide supplemental light, you really should provide that supplemental light when there is little sunlight available, because that allows the plants to use that light more efficiently. Traditionally, in the horticulture world, we've often made lighting recommendations based on the daily light integral. And if you just think about the daily light integral or the total amount of light over the course of a day, you might think that the timing of providing supplemental light doesn't matter, but it makes a really big difference.
Right. So how is the source and canopy measured ppfd across different spectral intervals of red, green, and blue under the artificial light conditions?
So you can probably answer that better than me, but the CIRRUS-3 has a quantum sensor built into the leaf cuvette that monitors the ppfd levels during these measurements.
Yeah, that is correct. There's two light sensors that are within the cuvette under the window that measures the light intensity from the light source or from ambient light when the light source is removed. So did you measure any chlorophyll fluorescence parameters under this experiment? So did they agree with the gas exchange data?
So we did not do chlorophyll fluorescence measurements, and that was partly on purpose, because based on my understanding, it's actually really difficult to get good chlorophyll fluorescence measurements if the color of the measuring light is very different from the color of the light that you're using to drive photosynthesis. So the CIRRUS-3 and most fluorometers use red light for measurements of chlorophyll fluorescence. I think the CIRRUS-3 does that. And so because red light cannot penetrate deeply into leaves, what you're basically measuring is largely the fluorescence coming from the upper cell layers. If you do that while you're using green light to actually drive photosynthesis, the measurement that you get with a red measurement light may not really be representative of the effects of that green light that is penetrating the leaf more deeply than your measuring light. So there are some real technical challenges there.
Right. One last question I think we've got so far is how confident are you that the sample size and resulting variability supports the conclusions related to the impact of green light relative to the red and blue?
Well, I'm pretty confident. So related to that, I didn't go into details, but I saw that someone else posted a question related to that. The way that June collected these measurements, we did that in a way that minimized the variability or maximized our statistical power. So she clamped the cuvette onto a leaf, and then she measured the leaf photosynthetic rate at a specific light level with 9 different spectral combinations. Then she went to the next ppfd level and again did the 9 spectral combinations until she had a whole light response curve for all 9 different spectra. And the reason that we did it this way is that we didn't have to worry about spatial variability of photosynthetic rates when you do leaf level measurements and you clamp the cuvette on one part of a plant one time and another part of the plant another time. She collected a complete replication on the same section of the same leaf, and because of that, that really helped us reduce variability and gives us a lot of faith in the results that she got.
All right. All right, well, I think that's all of the questions that we have. So I think no one else has any questions. I want to thank everyone for coming and participating. I'd like to thank you, Mark, for taking your time and giving this wonderful presentation. To let everyone know, any questions that we didn't get to, or if you have any additional questions, please don't hesitate to either call or email us here at PP Systems, and we'll share that with Mark. And would be glad to be of assistance to anyone that has any additional questions.
Yes, and in the emails that you all received from me, you can just reply to that email address with your questions. And as Mark said, he'll have a survey he'll be sending out to you that is very brief. If you wouldn't mind completing it, it just helps him going forward. And next week, early next week, I'll be sending out to all of you a link to the video of this webinar if you'd like to share it with somebody. So I guess that's it. Thank you, Mark. Thank you for attending today's webinar. To learn more about Dr. van Iersel's lab, visit www.hortphys.uga.edu. To learn more about Project LAMP, visit www.hortlamp.org. To learn more about Candidus Incorporated, visit www.candidus.us. And to learn more about the Cyrus 3 portable photosynthesis system, visit ppsystems.com.